My name is Shreyas and I'll be talking about how DNA is packaged in organisms. So, let's talk about why we're talking about DNA packaging first of all. What's the significance? Um, so, you know that all the genetic information that is passed on to children is passed on through DNA, and so that's a lot of information first of all, and this information is responsible for coding up all the proteins and all their functions. Everything is in that information. So uh, this lot, so much of this information is stored in the nucleus of a cell, and that nucleus is about 6 micrometers in diameter. So, and the total length of DNA is about 2 meters in each cell. So, yeah, that's like a lot of compactness for 6 micrometers. And we'll be talking about how DNA achieves such compactness. And just to give you like a real macro size view of that, the total length in the total length of DNA in the human body is about 6.2 billion kilometers, and that is about equivalent to 41 trips to the sun from the earth. Fascinating, right? So yeah, let's come to like the packaging aspect of DNA. So not like normal DNA or naked DNA is a double helical strand, which, yeah, it's basically just a double helical strand. So this strand needs to be somehow packed. Let's talk about a protein called histone. This is very crucial in the packing. So histones are basic proteins uh, which basically have DNA wrapped around them. So for 8 of these histones come together, and yeah, they basically form balls, these 8 histones, and DNA wraps around them. So this entire unit is called now a nucleosome. Sorry. Yeah. So this entire unit is called a nucleosome, and 2 nucleosomes are linked by something called linker DNA. So let's have a closer look at a nucleosome. So as I said, there are 8 histones total in each nucleosome. But there are only 4 types of histones present here. So each histone is present in a pair. Um, so one could question that why would DNA wrap around the histone? Like, it needs some kind of motivation, right? So the motivation is simply charge. You know that DNA has phosphate groups, and hence DNA is negatively charged. And these histones, so histones are basically proteins, and proteins are made out of amino acids, and a lot of lysine groups are present in these histones, which give them a positive charge. So yeah, so this positive charge and negatively charged DNA are attracted, and hence it coils around it. So yeah, that gives some stability to it. um, so this yeah, cool. So this kind of forms a bead on thread kind of structure. So these are the histones, and this is the DNA. It's like, yeah, these are kind of beads on a thread, but this is still not enough compactness. Like, the amount of compactness the DNA achieves is much more than that. So this actually coils into a more compact structure called the solenoid fiber. So basically 5 to 7 of these nucleosomes come together, and with the help of something called as the H1 histone, which is separate from the histones found inside a nucleosome. So with the help of that, they form like 5 to 7 of these come together and form this kind of structure. Uh, and when extended, they form a fiber called as the solenoid fiber. And yeah, this basically looks like a solenoid, so it's called a solenoid fiber. So yeah, that's that. You can see here that the DNA has uh, coiled into nucleosomes, and nucleosomes have coiled into a solenoid fiber. But that, even that is still not enough compactness. So we don't have like these solenoid fibers found directly in the nucleus. These solenoid fibers further coil into a structure. You can see it here, like it's coiling up, and this structure is finally known as the chromatin. And the diameter of this is approximately around 30 nanometers, and this is usually also known as the 30 nanometer fiber. uh, finally this 30 nanometer fiber coils up to form something known as a chromosome, and the chromosome is the one that is actually found inside a nucleus. So the destructuring sort of goes from this X-shaped chromosome to the chromatin fiber, to the solenoid fiber, to the nucleosomes, to the DNA. And histones are sort of like the helper proteins that are present along with it. So yeah, that is how DNA is packed so efficiently. So yeah, keep enjoying science, and goodbye for now.